Hello everybody, my name is Jordaens. I'm a PhD student at the University of Antwerp and a biomedical scientist at Novosanis. Today, I'll take you into the world of urine as an emerging liquid biopsy. The most often used liquid biopsy is blood, which uses either serum or plasma as a sample type. However, blood has several limitations that can be resolved by urine. Compared to blood, urine is absolutely non-invasive, can easily be collected at home, and does not add burden of substantial blood trials. Moreover, the collection of urine is not limited by the health status of a patient and does not entail any risk of transmission of bloodborne pathogens. The high volumes that can be obtained per patient address the limitations of low biomarker concentrations and allow for multi-omic testing to detect a complete panel of biomarkers. Let's first have a closer look at urine as a sample type and how it is used in research. First, there is a range of terminology used in the field to characterize the different times and fractions of the urine collection. First, there is first morning, being the urine collected at the start of the day. Then we have the first void, or first catch, or first pass, which is actually the first part of the urine flow at any time of the day, and mostly refer to the first 20 ml of the urine flush. And then we also have midstream, which is being the urine collected right after discarding this first part of the urine flow. And of course, some people also use random. This is actually urine collected at any time of the day and for any fraction type. Secondly, there can be a division made during processing. Most of the processing protocols will first centrifuge the urine sample to divide it into urine sediment at the bottom of the tube and urine supernatant on top of that. However, some protocols also work with full void, and this is then without a centrifugation processing step. Next to the different collections and fractions, urine contains a lot of components that can be used as biomarkers for the detection of diseases such as cancer, urinary tract infections, and a lot of other ones. Here, in this figure on the right, we can see that urine contains both cellular and cell-free components, but also proteins, metabolites, and volatile organic compounds can be found in urine, and even the concentration of metals in urine can be used as a biomarker. Urine clearly is an emerging liquid biopsy. A quick PubMed search showed already really fast that the last decade the number of publications concerning urine and oncology, or urine and liquid biopsy, urine and cell-free DNA, or urine and extracellular vesicles has increased massively. For urine oncology, for example, there was an almost doubling of the amount between the 3 decennia so far, which is amazing. Probably it will not be a surprise that urine can be used as a liquid biopsy for urinary tract-related cancers such as bladder cancer, prostate cancer, colorectal, renal, ovarian, and even cervical and endometrial cancer. However, urine can also be used as a sample type for the detection of thyroid, breast, lung, and pancreatic cancer, which are even a little bit more surprisingly than the first ones. It takes time to develop a clinical biomarker. The process of development of a clinical biomarker for early cancer detection is normally divided in 5 phases. Although many of our non-invasive biomarkers have already been reported, most of them remain in phase 1 or 2, a few in phase 3 and 4, and sadly none in phase 5. However, the clinical validity of any biomarker has to be evaluated for accuracy, reproducibility, reliability before they can become a clinical utility, of course. For the clinical application of biomarkers, the standardization of specimen collection, handling, storage, and the analysis of the biomarker is key for the quality assurance, the reliability, and, as expected, the reproducibility. With the significant advances in our analytical techniques and the tumor biology that we know of now, there is a great possibility of a more accurate, sensitive, and cost-effective non-invasive test for the early detection of cancer being developed in the near future. As you can see, there are promising possibilities for urinary biomarker research. However, there are still some issues that need to be solved for urine as liquid biopsy. These are actually really similar issues as the field is solving for blood. There is a lack of uniformity and standardization. The optimized preservation and storage conditions are not yet known. Optimized isolation and detection techniques are needed to be determined. And of course, validation studies in large cohorts still need to happen. Some of these issues can be solved by using a standardized and volumetric collection method, which exactly is what the Colli-Pee platform offers. Next to that, our platform also provides the possibility to choose a preservation chemistry based on your purpose. As you can see, we have 3 different series of Colli-Pee. Our small volumes, which captures 4 or 10 ml, our FV5000, which captures 20 ml, and our large volumes, which captures 45 ml. Next to that, we also offer all our variants in a neat variant without preservative, as well as with UCM or UAS, two of our available preservatives. Currently, there are also other chemistries in development to expand our portfolio even further and the capabilities that we can cover. But you have to stay tuned for more information on this via our website. So a little bit more zooming in on Colli-Pee itself as a unique, user-friendly device to collect first void urine in only 5 small steps: unscrewing the cap, mounting the tube to the housing, peeing, demounting the tube, and closing the tube again. The user can collect a standardized volumetric first void urine sample without interrupting the urine flow. Next to our Colli-Pee, to follow up during this COVID-19 pandemic, we at Novosanis also developed a postal kit to have like a more facilitation of home-based collection. Novosanis is active in 2 main application fields, namely infectious diseases and oncology. You can follow all our activities and current studies on our website. Today, I would like to zoom into one oncology study in particular. This study is URODETECT. This actually is my PhD study. It handles about urine as a sample type for biomarker detection for breast and prostate cancer patients. The results I have for you today are from our first pilot of our optimization study. Here, urine samples were collected from healthy female volunteers, healthy male volunteers, pregnant women, and breast and prostate cancer patients. With these urine samples, 3 different isolation methods were compared for their performance on first void urine samples, using qubit to measure the DNA concentration and tape station to measure the percentage of cell-free DNA. Next to this, all participants were also requested to complete a questionnaire regarding the usability of our Colli-Pee. So let's first have a look at the more scientific data. On the left graph, you can see the DNA concentration of the samples combined per isolation method. Here we can see that the 3 different isolation methods investigated performed comparable to each other. At least no significant differences were found. Although, based on the isolation method itself and on all the results combined, it feels like QIAGEN is a better fit. Then on our right graph, you can see the percentage of cell-free DNA of the samples of the different participant groups, which are colored per isolation method. And here we see that on average, 20% of the samples is cell-free DNA, which is, to our opinion, a quite good result for a first pilot study. Also here, no significant differences were found between the groups, nor between the isolation methods. So to conclude these data, we can state that for both the quantity and the quality of the cell-free DNA isolation from first void urine, all 3 investigated methods performed comparable. If we then secondly have a look into the usability data, I can share this with you because we asked the people via a questionnaire. The participants received several questions regarding the usability of Colli-Pee before, during, and after the use, as well as comparing the Colli-Pee and a standard urine cup, which we all know, of course. So on our left graph, you can see that most of the participants, about 71%, preferred to collect urine with the Colli-Pee over the standard urine cup. On our right graph, I've depicted the general experience of our participants. Here we see that about 80% rated Colli-Pee easy to even very easy to use in general. So to conclude these results, we can say that Colli-Pee scores really high on its user-friendliness and its easy handling. With this, I would like to conclude our presentation. Thank you for your attention, and if you have any questions, please feel free to visit our website or to contact me via the email address mentioned on this slide.